guys, welcome back. We're on lab 1.12 on population genetics, and we're going over exercise B in that lab. I already made a video for exercise A, so go ahead and check out my channel to find it if you need it. Okay, so we're looking at exercise B, and we're kind of doing something very similar here. And I will tell you in advance that I'm kind of cheating a little bit when I manipulate the lab, but it shouldn't have super drastic consequences. So we will just do the best that we can. Okay, so there's a couple of different rules here as we go through this. Now we're not talking about jackrabbits with their ears. We're talking about butterflies in Africa that have two alleles that determine color. So we're going to have, again, black beans representing the blue allele and white beans representing the tan allele. So in the species of butterfly, the blue allele is dominant over the tan allele. I did my post-it notes again for which allele, which is dominant, and then what I'm manipulating here. So like I said, I'm using black and pink paper clips. And I did the same thing for the tan allele and the recessive allele, which is going to be represented by my pink paper clips. Okay, so then it tells me to set up my initial population by placing 70 black beans and 30 white beans in a bowl. Again, I don't have time for that. And you probably don't either. So I'm going to use 7 black paper clips. 3, 6, 7, and 3 pink ones. And then record them as a percentage of the total population in decimal form in the parent population allele frequency column. So that is also already done for you. If you look here, we did 0.7 to represent the capital A, which should be in your bag, and 0.3 to represent the lowercase a, which should be in your bag. And then I already put your notes up here for what these mean. So we're going to, again, I'm just going to quickly explain how we fill those out. It's the same exact thing we did on the last chart. Now we just have different numbers to abide by. So again, to get the homozygous dominant genotype frequency, we're going to take our p-value, which remember again, the capital A is always representing our p-value, and we're going to square it. So p squared or 0.7 squared will give us 0.49. Okay, we have the heterozygous, which is represented mathematically by the variable 2pq. So we take 2, multiply it times the p-value, which is 0.7, and the q-value, which is 0.3. That gives us 0.42. And then we have our q-value, our q squared, which represents the homozygous recessive. And we take our a value, 0.3, and we square it. That gives us 0.09. So in order to find out our number in the population, again, that's representative of 100 alleles, which make up 50 genotypes. So we're going to take our frequency and multiply it times 50 individuals. So we have 0.49 divided by, times 50, not divided by. 0.49 times, I keep pushing the wrong button. There we go. 24.5. For the purposes of this lab, they kind of manipulated it for you. You're going to basically be rounding up or rounding down and try to eliminate the use of decimals, okay? In this case, they rounded down just because it's already such a high number of frequency for this homozygous dominant allele. And you'll see we'll get another decimal when we do homozygous recessive. And because it's such a small part of the population, we rounded up in that case. So there's our number of individuals. We can do the same for heterozygous. So we have 0.42 times 50 gives us 21, and then 0.09 times 50 will give us 4.5, which is rounded to 5. Okay, that's at your own discretion, and that's not going to make or break your lab. So don't stress about like which one you're rounding up versus rounding down. All right, there are different rules for pulling out of the bag for your observations, and you'll see that there is a surviving offspring here. So we're going to be able to fill that in later. So let's talk about how we fill in this chart and how we do our observed number. Okay, according to the directions, what we're going to do is select beans or clips out of my bag 2 at a time, and we're going to tally it up. So I made a post-it note this time again, except this time I did something a little bit differently. I made a list of homozygous dominant, which means that genotype is not going to be killed off if I pull it. If I pull a heterozygous, it's also not going to be killed off, and I'm going to make my tally marks here as I pull. And then for my homozygous recessive, if I pull that, I'm going to need to flip a coin. So that's where you need a coin from your materials list. I actually don't have an American coin in my office, but I have a foreign coin, so I'm going to be using that. Again, roll with the punches and do the best you can. If you flip your coin and you get heads, that means that your homozygous recessive individual is going to live. If you if you flip your coin and you get tails, that means your individual is going to die. Okay, so there's a lot to record here as you go through. And then it also asks you to record how many die, how many deaths you have. Okay. Okay, so then it tells you to do that for all of your charting. Let's go back to our data table and see what example observations they gave you. So now here, they say, again, remember when you fill this out, you'll have this first number from your tallies. So this is giving you an example saying that you pulled 23 homozygous dominant individuals, you pulled 22 heterozygous, and you pulled 3 homozygous recessive. And of those 3, it looks like 2 of them died because only 48 offspring survived. Okay, so that means 2 individuals in our population died. Again, that means that we flipped a coin, we got tails, and that means that they didn't survive. Okay, so these numbers are important, and I'll explain why once we get to this section here. So once we have how many lived, so we're subtracting 50 minus the amount of organisms or butterflies, in this case, that died for that number. We have our numbers tallied here for how many were pulled out of the bag, not how many survived or how many died, how many total were pulled out of the bag, because these numbers should always add back up to 50. And remember, to get these frequency numbers, we're just going to take what we pulled. So for homozygous dominant, we pulled 23, and we divide by the total number of opportunities for a genotype to be pulled, which was 50, because we had so many alleles, which would give us, oh, I'm so sorry. It's not 50 because we only had 48 survive. Only 48 survived. So we have 23 and we divide it by 48. And that gives us 0.479. We round that to 0.48. That's how we got that number. All right, for the frequency of the heterozygous genotype, we take the number that we pulled out of the bag and we divide it by how many in the population survive, which is 48. That gives us 0.458 or 0.46. And then the same thing for homozygous recessive. So we pulled 3 out of 48 that survived, so that gives us 0.0625, and we just rounded it to the 100th again. To calculate the observed allele frequency, they give you a, oh, I don't think I have it on our chart here, but I do have it pulled up somewhere. They do give you a, another equation, which is down here on your sheet. It's a little different from the first one. If you notice, in exercise A, we divided by 100. In this one, we're dividing by the number of alleles. So to walk you through how the math would work on that one, I'm going to go back to our data table just so that we can see the numbers, and I'll put my calculator up next to it. In order to get this number here, it tells us that we have to multiply for the p-value, or for our homozygous dominant allele. We need to multiply the number of homozygous dominant individuals in our population times 2. So 23 times 2. We add and account for the heterozygous individuals that have the homozygous dominant allele. So we're adding that to 22 in this case. And we're going to divide by the total number of alleles. And it tells you in the directions, the total number of alleles in the population is the number of individuals multiplied by 2. So in this case, the number of individuals that survived was 48. So that's what we're going to divide by. So that's what we're going to divide by for alleles. But if you notice, 48 individuals survived. Every individual has 2 alleles to make up a genotype. So to divide by the total number of alleles, we have to take this 48 and multiply that times 2. So 48 times 2 gives us 96, and that's the number we're going to divide by. So divided by 96 gives us 0.708, which is rounded to 0.71. Okay, so we're going to do it again. For the Q value, or the homozygous recessive allele, we are going to take the same equation, 2 times the number of homozygous recessive, which was 3. That gives us 6. We're going to add the number of heterozygous individuals to account for that lowercase a in that genotype. and we're going to divide by the total number of alleles in the surviving population. Again, there's 48 individuals. Each individual has 2 alleles to make up their genotype. So in total in a population, there's 96 alleles present. So we divide by 96. That gives us 0.291 rounded to 2.9. Okay, so here's a little different. They do want us to use our observed allele frequency for the next generation again. And notice the numbers are a little bit different. So if we were using beans like we were supposed to, we would now put 71 beans in our bag and 71 black beans in our bag and 29 white beans in our bag. But again, I do not have 70 and 30. So I'm going to keep it roughly the same. And so I'm just going to keep it as 7 and 3. My numbers could always change, right? My numbers could always be a little bit more change here in the end, at the end of this second line. Typically in this activity, what you're going to see is this number continue to increase and this number continue to decrease throughout the evolution of that allele frequency. And I hope that that's what you get if you do end up cheating. If you don't want to cheat and you want to get real results, by all means, go do that, okay? When you finish exercise B, you're responsible for filling out questions 3 and 4 in your lab report. So going back to our lab report, 3 and 4 pertain to exercise B. And then as you can see, there's only 2 questions left, and those pertain to exercise C. I will make a video about that coming up next. Hopefully that answers some questions for you guys on that one.